Hello, everybody, and today I'm going to be breaking down the Kruse Huberman Rubin podcast and giving you my analysis on that. And we're going to start by, in this video, part one, talking about optical signaling in the human body. So, without question, to me, the main point and takeaway of this talk was that light and sunlight is critical to human health. And furthermore, that optical signaling basically controls everything in the human body. And basically, the point is that cells are actually using optical signaling, photonic signaling, to control all of the biochemical processes. And one of the points that Dr. Kruse makes in other podcasts and his blogs is that there's over 100,000 chemical reactions happening every single second in every single cell of your body. And if you know any physics, basically the only thing that can control that is light and optical signaling. And so they talk about how we have these non-visual photoreceptors everywhere, both inside our brain and inside our body and on our body surfaces. And so he's kind of prodding Huberman, what's the point of this? And basically the point is because we use this optical signaling to control basically all physiological processes. And when you understand that point of view and that concept, then it makes complete sense why we have all these non-visual photoreceptors basically everywhere on our body, because we're sensing the light both from our environment, we're sensing the light in our environment and harnessing it for both energy and signaling. And then within our cells, deep within our tissues and cells, we're also using light to actually control all the biochemical reactions and metabolic pathways that are happening in us. So when we're talking about how organisms and human beings use light, there's two big kind of ways that we use light. So first of all, we use light to harvest energy, which is what we're going to be talking about first. And then secondly, we use light for signaling, which is what I'm going to be talking about second. So let's first dive into how we harvest energy from light, and specifically from sunlight. So enter the work of Dr. Gerald Pollack, PhD. He's at the University of Washington. So he's done some extremely important work on the interaction between light and water. And basically, one of the really big points in quantum biology that you realize when you read a lot of papers and listen to Jack Kruse's work and things of this nature is that water is basically where we store a lot of this energy. Dr. Kruse, I think even in this podcast, he talks about how we store a lot of photonic energy in the hydrogen bonding network of water. Water is basically a huge capacitor where we bury a lot of this light energy. And so basically what Dr. Gerald Pollack has done some very important work showing that red light, specifically infrared A light, water actually absorbs this spectrum of light. It's actually a chromophore for infrared A light. And when it absorbs infrared A light, it actually, quote unquote, structures the water. You may have heard of the structured water. This is exactly what that I was talking about. Now, this structured water actually has many names, and that's actually part of what makes it confusing. So it can be called structured water. Pollack also called it exclusion zone or fourth phase water. Dr. Jack Kruse and other papers in the literature that I read call it coherent domain or quantum coherent water, which is actually probably a better name for it. But that's an extremely important concept to realize, that basically what happens is when our cells absorb infrared A light, it actually does something to the water. It structures the water and basically separates out the protons and electrons, and it essentially creates a capacitor or a battery. And that actually allows us to move protons and electrons through the system, which we're going to talk about a little bit later. But this is an extremely important concept to understand. So additionally, our mitochondria actually are sensing and absorbing the light in the environment. So the mitochondrial cytochrome proteins 1 through 5 are all light chromophores. So essentially we have these cytochrome proteins. Again, we have 5 of them, 1 through 5, that are all on the inner mitochondrial membrane. If you want to see more about that, go check out my earlier mitochondrial videos. I go in deep depth and explanation regarding how the mitochondria works and explaining all this. But essentially, these cytochrome proteins in the mitochondrial inner membrane are all light chromophores, and 3 through 5 are red and infrared A light chromophores. And they work significantly faster and more efficiently when they're exposed to this red and infrared light. And even additionally, so cytochrome 5, which is the ATP synthase, which is literally a nanoturbine, it's literally a turbine that makes ATP. It can actually spin with just red and infrared light infrared A light exposure, even independent of food intake. So this is something that he actually talks about in the podcast. It's a really important concept to realize. And again, in my other videos where I talk about the mitochondria, in the earlier videos I've done on my YouTube channel, I explain all this. But basically, the health of the mitochondria is basically determines if you're healthy or not, how fast you age, and whether you have a chronic disease. And what's so important is that red and infrared A light actually makes the mitochondria work more efficiently, and basically makes them healthier. So when you are outside getting appropriate sunlight exposure, it actually improves the health of your mitochondria. It actually improves energy production and basically health and anti-aging, and helps prevent chronic disease. And so that's a really important concept that was touched on in this podcast. Again, the people that did a lot of that work was Dr. Doug Walls and Dr. Nick Lane. I talk about them a lot in my other videos, my earlier videos on mitochondria. Then lastly, as a kind of a bonus, what's interesting too is that three cytochrome proteins 3 through 5 actually absorb red and infrared A light, but the second cytochrome protein actually absorbs blue light, and that actually inhibits electron train transport in the mitochondria, inhibiting mitochondrial function. So this is actually the reason why, or part of the reason why, blue light can actually be harmful to our health. Continuing on with harvesting energy from light. So I want to talk about the role of melanin in harvesting energy from light. So melanin plays a huge role in human body, and in fact, he talks about mammals a lot. In this podcast, Dr. Kruse talks about mammals a lot. And basically, melanin has been amplified. This gene called POMC creates melanin, and then this gene POMC and melanin have been amplified in mammals to actually help us evolve and basically create the complex animals that humans are today, with the large brain that's allowed us to create this advanced civilization. So basically, melanin has a huge ability and plays a huge role in harvesting energy from light, and specifically melanin contributes to metabolism and overall cellular energy levels by absorbing light and then using that photonic energy to actually separate the nearby water molecules, creating the molecular hydrogen, 2 H2 molecular oxygen molecules, and at least 4 free electrons. Now, what's interesting is that the electron transport chain in the mitochondria, in order to make ATP, it actually runs on exactly this: oxygen, hydrogens, and electrons. And then additionally, basically, melanin creates all of the necessary constituents and substrates that you need to make ATP and run your mitochondria and run the electron transport chain. Then additionally to that, these free electrons that are created by the charge separation of water from sunlight hitting melanin, or light hitting melanin, these free electrons can actually be delocalized, and the hydrogens can also basically be superconducted throughout the system, throughout the cell, and throughout the fascial and tensegrity system to be moved wherever they are needed. And people have talked about this. Maywin Ho, Dr. Maywin Ho, PhD, has talked about this. Gerald Pollack's talked about this. There's literature. Some of the papers that are on grounding have talked about how electrons can be moved throughout the system. That's basically actually how grounding works. So that's another really important concept here, that we use sunlight to harvest melanin, or to harvest energy using the interaction of light and sunlight hitting melanin to charge separate water to actually create molecular oxygen and free electrons, which is exactly what the mitochondria needs to run the electron transport chain. And then additionally, these free electrons can be moved throughout the system to actually donate themselves, the free electrons, as antioxidants, and actually quench excessive inflammation and provide antioxidant and anti-inflammatory effects when they're actually moved throughout the system. So this is an extremely important mechanism to understand, and they go over that in this podcast. So now that we've talked about light and its role in harvesting energy, I want to talk about light with regards to cell signaling. And so first we're going to talk about circadian timing. So when we're talking about non-visual photoreception, so basically everything we're going to be talking about fits under this umbrella of non-visual photoreception. And again, that just means that these different molecules have the ability to absorb specific spectrums of light, different frequencies of light on the electromagnetic spectrum. And so basically, these different molecules, these different receptors that do non-visual photoreception and absorb different frequencies of light, they contribute to circadian rhythm timing. So you have these different photoreceptors that were talked about in the podcast and that are extremely important. So melanopsin is the main one. It's a blue light detector. It's on the skin, it's in the arteries, it's in the brain. It's a lot of different places. Our body is really basically loaded with melanopsin. And then you have neuroopsin, which is a UVA detector. It's on the cornea, and I believe it's also on the skin and in the brain. And then encephalopsin, which is a blue-green light detector, also in the brain. So circadian timing, basically these photoreceptors, they absorb these different frequencies of light in the tissues, in the cells, and then when they're absorbing that light, that's actually regulating their circadian rhythm, even independent of the actual eyes. So basically, we have two circadian signaling systems. We have one system through the eyes. So, during the day, you're normally getting blue light through the eyes, as well as UV light in the environment and red and infrared light as well. Really the full spectrum of light's present, during midday per se. And then as the sun goes down, less and less blue light present, and basically you're left with mostly red and infrared light in the morning and then at the end of the day. So that's actually regulating the central clock in the suprachiasmatic nucleus in the hypothalamus, and that's regulating the entire rest of the pathway through melatonin and cortisol signaling, etc. But all of your peripheral cells, so they're listening to the suprachiasmatic nucleus in the hypothalamus and the signals that it's giving out, but they also have their own peripheral clocks too. And basically, you want the peripheral clocks in the skin, say over your abdomen or over in your liver, the cells in your liver. You want all of these peripheral clocks to be yoked perfectly to the central clock. But however, what's happening in a lot of people in modern society is that we are blue light exposed through our eyes when the sun isn't out, say late at night, or even just to our skin. So then you're aging your skin more because basically your skin is receiving blue light when normally that wouldn't be out at night, and maybe you're wearing blue light blockers, which are actually blocking blue light through your eyes, but your skin's getting exposed to blue light. So you're basically getting a mismatching in signals, between the skin and the suprachiasmatic nucleus, and that causes accelerated aging and damage. So there's a lot of different ways that anytime there's any circadian mismatch that basically causes damage, and it specifically causes damage through mitochondrial function, because circadian timing is absolutely key for mitochondrial function. And the main point is that basically during the day you want your cells to be making energy, making ATP, and then at night you want them undergoing repair and undergoing mitophagy, and this is controlled by the circadian rhythm, which is primarily entrained by the light in your environment. And again, as I've talked about in previous videos and explained in great depth, the health of your mitochondria is the key to health, anti-aging, and disease. And so basically, again, just to recap this slide and this non-visual photoreception through these chromophores, again, melanopsin, the blue light detector, neuropsin, the UVA detector, and then encephalopsin is a blue-green detector in your brain. So these photoreceptors are present. So the melanopsin is present in the eye. The neuropsin is also present on the cornea. So these are taking in light from the environment that your eyes are getting, sending it back through optical pathways to the suprachiasmatic nucleus in the hypothalamus. That's regulating the circadian rhythm centrally, and then sending that message out peripherally to all the peripheral clocks in the rest of the body, basically mainly through the melatonin and cortisol hormonal signaling pathways. However, you also have melanopsin on your skin, and so that's also detecting the peripheral clocks. Basically, these peripheral clocks on your skin are also detecting the light in your environment. So if there's a mismatch, say you were wearing blue light blockers, but you're getting a lot of blue light on your skin, that can cause a mismatch in circadian timing for these cells. So basically, you want the light that you're getting through the eyes to the suprachiasmatic nucleus and then going out through the rest of the system, and the light that's on your skin, you want that all to be yoked perfectly with the circadian rhythm of the sun and of the earth. And that's what optimizes mitochondrial function. It allows for optimal energy during the day and then optimal repair at night. And that's ultimately what allows for the optimal health and vitality. So now I want to talk about light and cell signaling and neurotransmitter and hormone receptor interactions. So light in this podcast was talked about with regards to circadian rhythm timing, but it also plays a huge role in actually cell signaling and neurotransmitter and hormone receptor interactions. So non-visual photoreception happens not only through these photoreceptors that do circadian rhythm timing, but also through DNA. So Dr. Kruse has talked about how DNA is basically an electromagnetic strip. It actually absorbs photons and even just other parts of the electromagnetic spectrum, which actually affects DNA transcription. And so that's a way that optical signaling actually controls DNA and what proteins you made. And remember, in this podcast, Dr. Kruse basically talked about how basically all proteins are semiconductors, as Albert St. Georgi first hypothesized and Dr. Robert Becker proved about 20 years later. And then additionally, it's talked about how probably many other enzymes and other organelles and subcellular structures are also probably absorbing and emitting light, and it's basically controlling their function. So we know that collagen is also emitting light. And so we know that bone basically is emitting light. Again, Dr. Kruse in this podcast talked about bone being a light emitting diode. And therefore, we know that also all of the fascia in our body, especially when it is basically getting compressed or undergoing some type of mechanical deformation, is also emitting light. So again, and then the mitochondria, of course, is really the big one. So when the mitochondria is doing metabolism and making ATP, it is also creating biophotons, and that's really the big one, because melanin sheets are often basically right next to the mitochondrial colonies. And Dr. Kruse has basically hypothesized that these melanin sheets are actually harvesting the biophotons from the mitochondria and using it to charge separate the water to basically create these free electrons, as well as the molecular hydrogen and molecular oxygen, and actually creating energy and supporting metabolism through that mechanism, as I kind of touched on earlier. So that's another really important mechanism that light is affecting basically human health and supporting metabolism, and how light is really important. So last slide here, finishing up the role of light in cell signaling and neurotransmitter and hormone receptor interactions. So basically, what's a really amazing thing that Dr. Kruse points out in this podcast is that basically all of the big neurotransmitters and hormones actually have absorption spectra within the action spectra of visible UV and IR light. So for instance, dopamine, epinephrine, norepinephrine, and then the thyroid hormones T3 and T4, which all come from the phenylalanine tyrosine amino acid pathway, they all absorb 200 to 400 nanometer light, which is basically the UV range for light. So all of these very important molecules all absorb UV light. And basically one of the main points that Dr. Kruse is making in this podcast is that when you look at that and you see that these really important molecules all absorb UV light, first of all, that can't be an accident. Again, that comes back to the thing that Dr. Kruse talks about, where nature never makes a mistake. These really important molecules are all absorbing UV light. And then secondly, if it's not a mistake, like Dr. Kruse believes, that means that UV light is certainly not harmful for humans, we're led to believe, because these really important hormones and molecules are absorbing UV light, and it's actually affecting the way that they're doing signaling at the receptor when they're binding. So that's a really important point I wanted to make. And then when you look at these other molecules, melatonin, which both come from the amino acid tryptophan, and they also have a benzene ring, just like phenylalanine and tyrosine do. So the serotonin and melatonin both absorb about 200 to 320 nanometer light, which is exclusively in the UV range. And again, one of the main points of this podcast is that these molecules need to be programmed by this light to do their signaling functions properly. And if the molecule doesn't get exposed to the proper light and hasn't absorbed the proper spectrum of light, Dr. Kruse basically raising the question, does it work properly and do its signaling role properly when trying to bind to its receptor? And these are really important questions, at the cutting edge of quantum biology, that researchers, are trying to figure out. But basically, very few people are working on these problems. And one of the, whole big points of this podcast was that we need to get more PhD students and researchers working, on this project, on this problem, and on, looking at the specific role that the absorption of this nanometer light is having on it, and basically what are the effects of many people not getting this light on the signaling of these molecules. Is that contributing to the clinical symptoms we see in low dopamine because maybe the dopamine molecule isn't getting exposed to the adequate 200 to 400 nanometer UV light because you don't have enough UV light in the system, so then you're getting the phenotype of low dopamine because the dopamine molecule can't do its job correctly when it's binding to the receptor because the light signaling isn't correct. It's a real question and a real possibility and something that needs to be researched further. But when you look at the absorption spectrum of these molecules, it's basically certainly plausible that the light that they are absorbing is playing a role in how the signal is transduced at the receptor site. And if these molecules aren't getting exposed to the correct light, they may not be able to perform their signaling role correctly, which is my hypothesis and also Dr. Kruse's hypothesis and one of the points that he makes. And so again, I basically made a cheat sheet for a lot of the really important molecules and their absorption spectra that actually absorb light in this range here that we talked about. So we'll just go over it really quickly. But leptin absorbs, Dr. Kruse talked about, it absorbs light and appears to possibly affect the leptin signaling. And certainly one thing that is very clear in the literature that I've read is that circadian rhythm definitely affects leptin levels and leptin signaling. And if you don't properly regulate your circadian rhythm, you'll actually get leptin resistance, which actually leads to obesity, since leptin controls total body energy balance in the body. So that's another very important point that I think a lot of people will probably get confused about when listening to Dr. Kruse's podcast, but it's very important to understand why that's so important and why circadian rhythm is so important for helping with the obesity and metabolic health epidemic. And then again, regulated cortisol levels are actually regulated via UV light because cortisol levels are directly affected via the POMC gene because UV light cleaves POMC and can make ACTH, which is the precursor to making cortisol. So depending on the light you're exposed to, cortisol levels will vary. Then hemoglobin absorbs 250 to 650 nanometer light. Vitamin D is created via that 312 nanometer light, which is UV light. Vitamin A is created at 300 to 350 nanometer light, which is UVA. And sorry, I shouldn't say it's created, but vitamin A absorbs 300 to 350 nanometer light. Testosterone 200 to 240, which is actually in the UVC range. Estrogen absorbs 230 to 280, also in the UV range. And so again, you can see this point here. Essentially, all the major hormones, neurotransmitters, and signaling molecules have absorption spectra within the visible UV and infrared light range. And so light is playing a major role in controlling the fidelity and controlling the signal transduction of these molecules. And essentially, the hypothesis is, well, if you're not absorbing this light and these molecules can't absorb this light, is the signal still as effective, when the molecules binding to the receptor? And the hypothesis is that if you're not absorbing this light, then the signal transduction might not be as effective, and maybe you can get things like the phenotype of a low dopamine. And so that's the hypothesis that's being put out there, and I think it is something that certainly needs to be researched more. But also I think people need to consider, trying to get more optimal sun exposure in order to maintain health and in order to maintain the optimal signaling of all of these molecules and the effects that they have on a healthy body when you're exposed to the proper light. So that brings me to the end of this presentation and to the end of my breakdown of the Kruse, Huberman, Rubin podcast, my analysis part 1. Thank you so much for watching, and if you liked the video, please feel free to like, comment, subscribe, as that really helps support the channel. And then lastly, I just want to mention that this video presentation is purely for educational purposes, and this is not medical advice. Thank you.